All right. Thank you very much, James, for agreeing to give a Build a Cell seminar. And go ahead.
All right, brilliant. Thanks, Kate. So yeah, so it's great to be here today as part of the seminar series. And yeah, so a little bit about myself. So yeah, I'm Dr. James Hindley. I'm the Department Fellow at the Department of Chemistry at Imperial College London. And I'm part of the Membrane Biophysics group. And we're sort of a collective of kind of, at the highest, kind of 40, 50 researchers, super interested in sort of fundamental and applied membrane biophysics. And obviously one of the kind of topics in the area, and something that we're all, everyone on this call is really interested in, is how can we actually, you know, use our understanding of sort of molecular interactions, of physics and chemistry, and how can we kind of use that to make biology? And so this is an increasingly big part of our research in the group. And today I'm going to talk a bit about my and some other researchers, some other students' research in how we can actually engineer synthetic cells that can sense and respond to their environment. So I'm going to start by kind of thinking a little bit about bottom-up synthetic biology and top-down, and you know what's really driving our bottom-up approach. And so on one hand, we have thinking about complexity. We can think about transport systems. So we can see on the left, this is just the regular transport system, the subways and the train links and all of this. And this is obviously a pretty complex network, but this really pales in comparison to the complexity of, this is just a part of the cellular metabolic network that we find in cells. And you know, if we're going to take one part, so you know, thinking about the transport system, well, if one of those stations goes down in that network, well, that's obviously going to affect the flux of movement through it. So that could be subway trains, it could be cars, and it's exactly the same in a metabolic network. As we start to manipulate it, well, if we manipulate one node in that network, it's those changes are going to cascade through, and it might be hard to predict some of those changes. So the bottom-up approach obviously allows us to construct simpler systems where we kind of can more easily understand how these systems interact. And as we start to build complexity into it, we hopefully still have control over it. So these are much, I would say, easier systems to engineer compared to living biology. And so I won't spend too long on this. I think a lot of people on the call are kind of aware of themselves. But you know, our approach to, you know, what is a synthetic cell is really how can we kind of take artificial components and can we replace all of the components that we find in a living cell. So for example, an artificial membrane to represent the biological membrane, in vitro transcription translation to you know, to allow us to generate proteins, pores to allow connectivity, and you know, how ultimately do we take sort of natural and engineered components and bring them together in defined ways so they can perform interesting and useful functions. And as a challenge, you know, we really generate complex behavior using a small subset of molecules, you know, an absolutely tiny number when we compare it to a living cell. And so by behaviors, I'm really thinking about these kind of archetypical things that we think of something that's alive. We think of things, you know, like it can move around. We have the classic thing here of a phagocyte-facing bacteria. Communication, so not only cells talking to each other, but how does a cell, you know, integrate? How does a cell kind of respond to a signal in its environment? How does it actually, you know, use that signal and have an output and a change in its function? And then finally, you know, adaptation. So how does the cell adapt and thrive in different environments? I'm going to talk about some simple systems that we've built that can demonstrate behaviors such as motility and communication. And really the heart of this, unsurprisingly, as I said, I'm from a membrane biophysics group, is lipid membranes. So one of the systems that we use most commonly when we're thinking about building synthetic or artificial cells are lipid vesicles. And so these are obviously well used in the community as the, you know, our foundation or the chassis for a cell. And I'm just going to highlight a few of the vesicles that we can build. So we can build them across length scales now, anywhere from the sort of 50, 100 nanometer length scale. And this has kind of been used for 30, 40 years in drug delivery. We can actually, where we can make nanoscale compartments. We can encapsulate content within the aqueous pore or the hydrophobic membrane itself. And so this has been used, of course, to change the sort of pharmacokinetics of drugs and to deliver them to selected locations in the body. But over the last 10, 15 years, what we've really done as a field, or I should say multiple fields that are kind of interested in these soft materials, is we can now make these not only across kind of length scales and controllably, we can control their size, but we can also control the compartment number, and we can control the connectivity between them. And on the right, that's just an optical micrograph of a 3-compartment vesicle, where the connectivity between those compartments is defined by that shared internal membrane. And the model that I'm going to talk a bit about today really is this kind of vesicle-in-vesicle system. So we like to call it a nested vesicle or a nested synthetic cell. You might have heard the term VESICLE as well. And I just want to spend a moment to talk about this because I think it's increasingly being used by our community. And one of the great things about it is kind of the simplicity in how we can kind of assemble these in a facile way. So because all we need to do to form these is to encapsulate at least one type of compartment in another. Normally, when we do this, we're encapsulating nanoscale compartments in a microscale sort of GeV chassis. Because we're using techniques such as phase transfer, we can, in parallel, to encapsulate many different compartments alongside each other, and so we can rapidly build the complexity of our synthetic cell. And so I'm going to kind of come back to this a couple of times throughout the talk. You can see how in our group we're trying to sort of push the boundaries of what we can do with this system, and hopefully at the end of the talk you'll think that this is maybe worth pursuing in some research projects, worth using. So we have lipid membranes, and we can actually make them, can form different architectures. But also, how do we go beyond using the membrane as a chassis, as an inert kind of compartment? Well, on the right, I've highlighted some ways in which we can do this, and this involves kind of using kind of chemistry, biology, and biophysics. So, for example, we can, thinking about synthetic chemistry, well, we can utilize functional groups that might be photoresponsive, or redox responsive, or pH responsive, and then by modulating the environment that the vesicle is in, we can start to modulate things like permeability and shape. We can also, of course, use the wealth of molecules that we get from biology. So in our case, we like to use ion channels and nanopores, but of course you can start thinking about conjugating enzymes. And, when you're thinking about in vitro protein expression, then we can start to express all kinds of proteins within a vesicle that either interface with or are actually integrated into a membrane. So then finally, we can use membrane organization. So, we understand, because of so many sort of pioneering works in membrane biophysics, we understand for more complicated vesicles, so let's say vesicles of 2, 3, 4, 5 lipids, we start to understand their phase behavior. And often what we find is that we see lateral phase separation, where instead of just a single phase of a membrane, we start to see 2 or 3 phases. And by controlling that, we can start to control the localization of content in a membrane, and also can often modulate the permeability. And so we've used a kind of a variety of methods in the last couple of years. And that's really driven quite a few research projects, some of which I'm showing here. And today I'm going to highlight 4 of these. So the first is how we can use mechanosensitive ion channels to build kind of signaling pathways in a nested vesicle cell model. Secondly is how we can use a similar architecture and use photoresponsive functional groups. And this allows kind of user control over a biocatalytic reaction. Third is how we can then start to dynamically reconfigure the subcompartments in a nested system. And then finally, how we can start to think about interfacing lipid vesicles with membrane-free systems or membraneless compartmentalized systems. In our case, how we've used them to stabilize and control the motility of dextran-rich droplets in peg-rich phase, phase separated system. Okay. Yeah, so first, or finally, in terms of, I guess, maybe final introduction slide, I want to highlight that one of the great things when you're trying to build a cell from bottom up is this plug and play approach. Lots of people have referenced this, variations of it. And, we can actually start to build by using just different molecular parts, whether from biology or synthetic chemistry or nanotechnology. We can bring things together in contexts that you just don't find in school. And so here, just thinking about biochemistry, I've got 5 molecules on the screen. Most of these are sensors, and they're sensors of different membrane properties, such as the bending rigidity or the spontaneous curvature of a system. And so if we actually take a membrane sensor and we combine it with an effect, often things like enzymes, well, we can start to build up controlled interactions between these. And so just to highlight that, I'm highlighting 2 on the screen at the moment. We can see on the left, we have MSCL. This is the mechanosensitive channel of large conductance, and this is really a sensor of membrane tension or asymmetry. And what we've showed in some of our previous work is we can combine this with phospholipases, which can generate asymmetry, and we can use this to sort of build protein interactions that occur through a lipid membrane. So, just to talk a little bit more about MSCL, this is a bacterial membrane protein, so it was originally found in E. coli, and it allows E. coli to survive hyperosmotic stresses. So when the membrane tension increases, MSCL will open, and it will form, for a biological pore, a very large 3 or 4 nanometer pore in diameter. So lots of content, even small proteins, can go across this. And so what we were interested in is showing how we can actually utilize mechanosensitive channels, MSCL, put them into a lipid vesicle and generate a mechanosensitive material. And so this is quite a few years old now, but in this work, we can see, well, in this cartoon, we can see MSCL that's reconstituted into some lipid vesicles. We can see it reconstituted into the membrane. And then we have a cargo encapsulated in there. So in our initial work, this was a small dye, but of course it could be a drug. If there's, in the external environment, if there's phospholipase present, or phospholipase combined to the membrane, it can cut single tails in this lipid membrane, and it only cuts this in the outer phase. So this is generating lipids in an asymmetric manner. This membrane asymmetry is sensed by MSCL, and we can get release of cargo. And so we're actively trying to use this for drug delivery approaches, trying to exploit high levels of phospholipases that you might find in diseases such as cancer. But also what we're really interested in doing was using this to control, let's say a signaling pathway-based type system inside artificial cell. And so what we can see here is our approach to this, which is where we actually take MSCL vesicles and we encapsulate them alongside phospholipase into a giant vesicle chassis. But the key to this is actually controlling this interaction. Obviously, we don't want phospholipases kind of going around and cutting up membranes without, with no control. And so we, fortuitously, phospholipases are calcium dependent. And so our approach was to co-encapsulate these into a nested system alongside chelators such as EDTA. And so EDTA will ensure that there's no free calcium in the vesicle, and so we don't have any phospholipase activity. If we then porate this external membrane, then we can control the influx of calcium into the synthetic cell. this calcium will then activate the enzyme, which can start to produce single-tailed lipids in the internal compartments here. MSCL can sense this. And so we actually end up with content release into the lumen of the vesicle. And so we wanted to sort of take a step-by-step approach. First of all, confirming that this phospholipase membrane MSCL kind of protein interaction could actually control content release. We showed on the right here that actually we see phospholipase dependent release of a calcein dye. And this dependence we only observe when we have MSCL present in the lipid membrane. And so this sort of reassured us that that we could use phospholipase to trigger flux through a protein channel through MSCL. And then we went on to confirm that we can actually chelate calcium and we can control the activity of the enzyme. And so here we show that as we increase EDTA above, so once we have an excess, we completely turn off enzyme activity, and we can show this because we completely stop calcium flux. And then if we add an excess again of calcium, we can reactivate this system. And again, we show kind of stronger reactivation as we have a larger excess of calcium. So then we've assembled the full system as shown on the left here. And we monitored this using fluorescence microscopy and spectroscopy. I'm going to show the microscopy data here, but if you're interested in more of the data, you can find it in our publication. And here we just show in red that when we have MSCL present in these internal compartments and alpha-hemolysin present in the external membrane, only when we have both do we actually see activation of this pathway, and we can see calcein. I should say this is a dilution of calcein in the GV lumen. So as the calcein dilutes, fluorescence increases, and we see this when we have both membrane proteins present. If we remove either of these in yellow or blue, we pathway activation. And so this was a fantastic proof of concept. But what we're really interested in doing now is kind of utilizing this as the core of a signaling pathway that we can operate and use to kind of in different applications. So we're interested in thinking about the inputs and the outputs. In terms of inputs, well, at the moment, we just use alpha-hemolysin, which kind of permanently opens that outer membrane. So, of course, we're thinking about, can we actually make this a gated process? How can we do this? Do we need to think about other proteins, or can we actually think about using lipids, or even, not using lipid membranes at all and thinking about more porous outer membranes, polymeric membranes, perhaps? In terms of the outputs, well, we're interested, of course, in things like drug release, but also using this to control parts of a protein, in vitro transcription and translation processes. And also, can we actually turn this off? Can we generate feedback into the system? If we can generate feedback, I think this becomes much more useful, and obviously it becomes closer to biological processes that are on demand and not just one way. Okay, I'll take a kind of a step sideways now and talk about instead of building systems that can respond to their local environment, how can we actually, again, use sort of stimuli-responsive membranes to generate user-controlled synthetic cells that have a high level of user control over the processes that we're building into them. And so in order to do this, this is kind of not using any biochemistry whatsoever, but actually just using synthetic chemistry and sort of implanting functional groups into lipids. So here we can see two lipids. There's EPPC, which is a saturated phospholipid, and importantly, at room temperature, it forms very rigid membranes. And if we combine that with a light-responsive lipid, DC89PC, well, in each of the hydrocarbon tails of this lipid, we see this dialkyne functional group. And so, again, if we combine these two lipids together, what we get is like a phase separation between them. And this is really based on the fluidity of the two lipids at room temperature. So DPPT forms very sort of rigid membranes. DC89PT is more fluid. And so what happens here is we get a sort of a concentration of the light responsive lipid in these small domains. And this means that when we shine a UV light on it, we get a really high efficiency of photopolymerization. And as what happens under UV light is we get cross-linking of these alkyne functional groups into longer polymers. And this affects the permeability of the membrane. So we can actually form transient pores that form as this photopolymerization is occurring. And we wanted to show as a proof of concept that we could use this to control a biochemical reaction inside a nested vesicle. So this architecture is very similar to the previous project, except instead of in the internal compartments using MSCL, we now have these photoresponsive membranes. And encapsulated inside those is a FDG, which is an enzyme substrate for beta-galactosidase. So we have beta-galactosidase co-encapsulated. And if we shine a UV light on our system, we can release FDG into the main compartment of the nested vesicle. And then this is catalyzed to a fluorescent product, which again, we can monitor with fluorescence. And so we just use fluorescence microscopy here to show, again, proof of concept that we can control these reactions. And where if we irradiate our vesicles for 10 minutes, and then we leave it in a further 30 minutes for catalysis to occur, we see a strong fluorescence in the vesicle. And if we don't apply any UV to our system, then we don't see any response. And so we're super interested. Well, first of all, I should say, I think this is a nice example of how we can start to think about building orthogonality into the synthetic cells we're making. And one way, of course, is to try and use a stimuli that biological systems don't normally respond to. In this case, it's light. I realize, of course, that some light is used for different organisms, but you know, if you think about the context for the synthetic cell you're building, then we can try and think about, again, orthogonal stimuli to the stimuli that natural cells respond to. And so we're interested in using this, of course, to control chemical reactions, and we're interested in using different sort of molecular motifs that are more biocompatible to then integrate this alongside, you know, various other projects, and eventually to start communicating with biological cells. And something that one of our PhD students, Ignacio Gisbert, is doing now, and hopefully we'll have that published. Third, I want to talk a little bit about, so nested vesicles, not only can we make them, and we can make these in quite a kind of facile manner. These are pretty simple to produce compared to some cell models. But one criticism of them, of course, is the connectivity between the internal compartments. And it's very hard to control that. So one thing that we've been interested in is starting to do this. Can we actually control the spatial organization of a nested vesicle? And this project was really led by Greta Zubates, who's just finished her PhD in the group. And Greta showed that by utilizing electrostatics, that we can control the association of the internal compartments with the external GUV membrane. And importantly, that this association is reversible and does not destabilize either of these membranes. And so Greta used positively charged POTAP and negatively charged DOPG, using a small amount of negatively charged PG in the internal nanoscale compartments, and then put the DOTAP into the UV membrane. And this allowed her to build various different architectures. First of all, we showed that if we're by encapsulating calcein into the internal compartments and rhodamine, by encapsulating calcein into these internal compartments and also labeling the membrane of these internal compartments with rhodamine, that we can show that these bind, and that this binding doesn't affect the permeability of either of these membranes. So the calcein is localized, it co-localizes with the rhodamine, and it isn't spread throughout the vesicle. So we're confident that this is an association and not a destabilization of the membrane. And so then Greta used, again, rhodamine and Cy5 just to show that we can localize these membranes onto the inner face of a GEV. We can also localize them to the outer face, and we can start to layer these on in like a layer by layer approach. And then we can also actually bind vesicles to both faces. And in terms of this externally localized architecture that we show here, we like to think of this as potentially in the future, using these synthetic cells as kind of depots for the release of extracellular vesicles. So we could think of the communication between synthetic cells mediated by these external compartments. So Greta went beyond just using charge to bind internal to external membranes, but again started to use phase separation as a tool to further localizing them. And so what we did here was effectively concentrate the positive charge in the GEV membrane into patches on the membrane surface, and specifically into the liquid disordered domains. This GEV now has 4 lipids in it, and wherever the membrane is yellow, it is this liquid ordered domain, and then this sort of holes in fluorescence. This is where the positive charge is localized. If we then took SUVs, our sort of external smaller compartments, and we labeled these in blue, then we could actually show that we get association of these smaller compartments only to the liquid disordered domain. And so this again shows that we can not only control in a simple way the localization, but we can start to get kind of greater and greater control over the process. So beyond setting up, but beyond controlling compartmentalization, what we showed is that actually we can modulate this, and we can modulate this in a stimuli-responsive manner. So first of all, we can start to control the iron or salt concentrations in our local environment, and that these synthetic cells can respond to them. So as we increase salt, we start to dampen the electrostatic interactions between membranes. And so we can take internal compartments that are bound to the GEV membrane. We can increase salt, and then they will kind of drop off into the lumen. And we show this here, having, again, very low concentrations of calcium chloride. We can see by looking at the cross-sectional fluorescence of our system that the fluorescence is localized to the edge, to sort of the GEV interface. And then if we increase salt, well, then we actually get this fluorescence that is kind of homogeneous throughout the lumen. Again, if we reduce the salt, we can actually get relayering. And I think the key thing to stress here is that I'm not just talking about, for example, encapsulating salt into the vesicle and that affecting the ionic strength. Actually, what we see is that when the external salt concentrations change, we can affect the internal compartmentalization, which is really quite an interesting behavior. And we think that this might be happening because of maybe a change in membrane capacitance or a change in membrane tension that is affecting these, essentially an external change affecting the internal membrane organization. So that was one way in which we wanted to sort of probe the system. The second is actually, we wanted to see if these had any sort of mechano-responsive behavior. In order to do this, what we did was we would tether a vesicle to a surface, we bind it to a surface, and then we would use an optical trap, a laser, to effectively pick up the vesicle and pull it from the surface. As we pull it, the vesicle remains attached through a membrane tether, and at what we noticed was at a critical tether length, we saw disruption of these of the organization of the system. So again, we have inner nanoscale compartments that are bound to the GeV surface, and beyond this critical tether length, these would drop off into the lumen. And we again showed this using the cross-sectional fluorescence of the system. And so now what we have here is actually showing that not only can we respond, not only do we have a system that can respond to where the entire population can respond to an environmental change, but we actually have a system that can respond mechanically. And we're really interested in sort of understanding this further. Again, we think that this is quite possibly occurring due to changes in membrane tension that occur as we pull the GeV, and as that GeV membrane runs out of excess membrane area, we start to cause that drop off or that loss of organization. And we're interested in also, of course, using this to control the onset or rate of chemical reactions within the next system. Okay, so finally, how about we get rid of the external membrane? So I've talked about this in detail for pretty much the whole talk, but how about we start to think how we can use vesicles and interface them with membraneless organelles or membraneless compartmentalized systems? And the system that I'm going to talk about are these aqueous two-phase separated solutions, in particular PEG and dextran aqueous separated solutions. And so these occur where if you take, for example, PEG and dextran polymers, particular molecular weights, and you dissolve them in solution, what can happen at particular concentrations is that you get this phase separation. And this is, again, this isn't oil and water or anything like this. This is completely water-based. And we can shake these up and we can form emulsions with them, but of course we'll get that separation in time. And so what we thought we could try and do, inspired by a great paper by Christian Keating lab, is try and stabilize these emulsion droplets with liposomes. And critical to this is the composition. So what ends up working really well at stabilizing droplets is having negatively charged vesicles or liposomes that also contain pegylated membranes. So what we found is that we can get a strong attraction of these vesicles to the interface, to the dextran-PEG interface, only when we put this pegylated lipid into the membrane. And we found that around sort of 1.5% we got optimal stabilization. If we don't have this PEG lipid in the membrane, you actually get localization of the vesicles into the dextran-rich core. And this behavior is also observed with biomolecules. For example, the larger the molecular weight of an enzyme or a protein, the greater the localization in dextran-rich phases. And so we wanted to show, similar to Greta's work, that we could, first of all, we can stabilize these systems, but also that we are stabilizing them with intact liposomes. So again, we have calcein loaded liposomes that also have a rhodamine stain in the membrane, and we see this this overlay of the calcein and the rhodamine fluorescence, and the calcein fluorescence importantly is localized to the interface. So these are stable liposomes. They're not forming some other kind of lipid layer. Okay. I should say this work was really led by Xiaobin Zhang, who's again just finished PhD in our group. And what we did is we took these droplets, and what Xiaobin found out was actually that these droplets could respond to a PEG gradient. So in order to generate a gradient, we would have in our observation well our stabilized liposome stabilized dextran droplets in our PEG-rich solution. And by adding a water source on one side of the well, we found that we actually observed this really interesting motility. So I'm going to play this video. And so what we saw is droplets moving towards the water source But critically, this movement only occurred after a certain extent of rearrangement of our liposome stabilizers. And so in order to understand this, we thought about the interfacial tension gradients that we're really setting up when we add water to the system. So when we add a water source, we effectively form a PEG gradient with a low PEG concentration of the water source and high PEG concentration away from it. And this gradient sets up a Marangoni flow, which causes the redistribution of liposomes in these stabilized droplets. As these liposomes redistribute, well, this exposes droplet surface area, and you actually form an opposite interfacial tension as more and more of the droplet surface area is exposed. And so once this goes beyond a critical surface area, these two interfacial tensions cannot balance, and so you actually get movement of the droplets to the water source. And here this occurs roughly when over 60% of the droplet area has been exposed due to liposome redistribution. And so we're really interested now in trying to use this as a kind of a novel cell mimic and see what kind of, whether we can start to increase the complexity of these systems in terms of their internal contents, and again see how we can utilize the chemotactic behavior of these, along with, again, things like the ability to produce proteins or release drugs and so forth. Okay, so I'm just going to conclude that. And I'd just like to stress a few kind of final points. The first is that we can use, elements of functional chemistry or an understanding of biophysics, or just, the wealth of biomolecules that we can take from different organisms, and we can make responsive lipid membranes just by using one or two of these. So a lipid membrane doesn't just have to be a chassis, it can be an active part in a cell, just as in biology. Secondly, when we start to integrate these, let's say, motifs with vesicle engineering techniques and building different architectures, we can start to build cell models that have functions that kind of go far beyond the number of molecules that we're active. And then finally, these cell models can demonstrate behaviors like communication and motility. And hopefully this is kind of the start of, now we've generated interesting behaviors, we're interested in thinking about how we can use them. So for example, the signaling pathway I highlighted, we're interested in trying to implement that into cell models that can then respond to biological environments and potentially be used in applications of delivery or biosensing. Okay, I'd like to thank all of the sort of membrane biophysics group, including the people listed here, our fantastic collaborators, our funding sources, and all of you for listening. So thank you very much, and I'm happy to take questions.
Thank you so much, James. That was awesome. There's a lot of questions in chat, and because of my upcoming prelim, I have to hand it off to Carlise for moderating the discussion. Just to remind you, if you could read a question in chat before you answer it, that's going to go on the recording.
Okay, so you want me to read it out loud first, or?
You do, or yeah.
Yeah, okay. All right, so I'll just kind of go through the chat questions. Okay, so there's one from Teresa asking, does the activity of mechanosensitive channels depend on the composition of the vesicle membrane? Or are they universal, and will they work in most stable lipid membranes? Yeah, that's a great question. So what we found is that they will work in, let's say, just a DOPC or a POPT membrane, a very simple single lipid membrane. But we see increased activity in our case when we use negatively charged lipids. But it depends on the context as well. So there have been other studies that have shown that the protein doesn't require particular lipids to function. So it might be that in our case, what we're seeing is affect the way we're putting the protein into our sort of model systems as opposed to a biological membrane. But yes, they are pretty universal. They will, there's examples of them functioning just in DOPT or POPT membranes and effectively working as sensors of membrane tension, the local environment. As if you have osmotic gradients, then you can activate these proteins in very simple lipid systems. Secondly, by Peter Bandel. Sorry if I missed it. Are you expressing MSCL in vitro or is it purified? Yes, so I didn't specify that. We purify from bacteria. So everything here is purified recombinant MSCL, a mutant of it that we can chemically gate. And we often reconstitute it, but we have also done some more recent work where we express it in vitro. And then, Alan Liu and Vincent Noiro have some published papers on that. So it's been looked into before. From Brendan, would you speculate that polymeric membranes or some mix of artificial polymer with lipids would be at the end of a synthetic cell than pure lipid membrane? That's a really good question as well. I think it really depends on the application that you're trying to think of. So I think in applied science, it might be that actually some blend of membrane would be better for a particular application. It might be more stable. But if you're thinking about a replicating system, then it's going to be harder to obviously generate a replicating system as polymers and lipids. Anna Marie asked, how stable are the DOTAP and DOPG vesicles? Could you expect proteins inside it? And this is cool, thank you. Yeah, thanks, Anna Marie. So the vesicles are reasonably stable over a couple of days. In terms of the nested system, we haven't tested the long-term stability, but I believe it'll last kind of overnight. So it's long enough to express proteins in it. We've never tried any sort of in vitro transcription translation in those systems, but I imagine that it would be reasonably compatible. So from Karen, saying, could you make vesicles inside vesicles with the internal vesicles being 2 or more? Yeah, you can absolutely do that. If you have a method to encapsulate things in parallel, like phase transfer, whether that's microfluidic based or just in an Eppendorf, you can just have 2 internal populations, you just mix them into the same aqueous solution, you do your phase transfer. How big can a liposome be and still remain stable with the vesicles inside it? Yes, so can you have one of those larger GV stuff with internal liposomes? I mean, in theory, you can make liposomes that are, 100 microns or maybe even bigger. As long as the internal compartments are not interacting and not destabilizing the larger one, I don't think the internal contents will really affect the liposome stability at a certain point. When they're big enough, I think they'll just start to fall apart based on the size of them. So yeah, I don't think it depends on the internal content so much, at least with the systems that most people use. It's just a matter of the larger compartment stability. And then Michael asks, can you use other types of channels? Absolutely, and it's something that we're super interested in, is just using things like ligand gated or voltage gated channels. The issue with some of these is you either have to make them recombinantly and put them into a membrane, which can actually take quite a lot of time. There isn't a universal way to do that. Each protein needs to be treated individually, or you work with protein transcript, protein expression systems, which different groups have worked on, but are not too many channels. Again, when you're working with membrane proteins, it's a lot more complicated than working with GFP or other soluble proteins. So you need to be confident that what you're making will be able to fold and integrate into the membrane. So I think that has put off some people, some groups working with that, but I wouldn't be surprised if that's something that comes out soon, or is used more regularly, especially when there's a good protocol for it. Okay. I don't know if there's any other questions. If there are, I'm happy to answer them.
Yeah, so it sounds like we've kind of gone through the chat, but feel free to unmic and ask a question if you have one now towards the end. So I'll wait a couple seconds for anybody to fill in either with their mic or in the chat. Give you a couple seconds to round off those questions or follow-up questions.
Hi, I have a question.
Hi.
Hi, thanks very much. That was really amazing. And I'm coming more from the engineering side, to be honest. So it was really helpful to have all those details, but I'm quite interested to know, you know, realistically, how far away do you feel that these approaches you've mentioned are likely to be able to be done in high throughput and actually be used as like commercial alternatives in these applications? Yeah,
for sure. That's a really good question. You know, when I think they are more complicated. Obviously, the more complicated the system you're trying to build, the structure you're trying to build, the harder it's going to be, because there's just all these parameters that you're trying to control. Throughput is a big part of that, but there are, for example, microfluidic approaches to building all of the structures that I've showed here today, that some of which we've developed, and then there's other methods for making, giant vesicles on the chip. One of the great things when you do that is that, they're all the same size, so they become more engineerable straight away. And because you're making them in flow, it's not batch based like all the things that I've highlighted, and you can, in theory, start to scale them up more. Microfluidics comes with its own challenges, but it is something that I think more and more sort of startups are using microfluidics to build different products, droplet-based products. And in this case, I wouldn't be surprised if there's companies that are looking to use similar approaches to build vesicles. In terms of the things that I've highlighted today, again, I would argue that the more proteins you're putting into something, often the more expensive it's going to be, and potentially the less stable it's going to be. So one of the good things about kind of a plug and play approach is that you can choose either proteins that are stable and use a small number of them, or you can actually just try and create function from either synthetic chemistry or again, just things that are a little bit more robust and therefore I think a bit more applicable for short-term translation. So I guess that's a long answer, but I think some of the systems are definitely closer than others, and generally if it's a bit simpler and it doesn't require lots of sort of biological components, it's probably going to be easier to translate that.
Cool, thank you so much.
Thank you. Okay. I don't know if there's anyone else.
Wow. Last chance for
everyone.
Questions. Well, I'd like to first thank you, James, for sharing your wonderful work with us. It's amazing. I love it. And I'd like to thank everybody else for attending. This is what makes these seminars possible. We still have people that are attending regularly, and that's good. We got to get more people on board with that also. So
thanks, Felice. Yeah, it was great to chat, and thanks for all the questions. And if anyone has any questions after this, then by all means please email me, or if you want to chat about anything today, just let me know. I'm happy to speak about collaborating or just talking about science.
Wonderful. All right, well, with that, we will conclude our seminar for today. Thank you, everybody.
Thanks, everyone. Bye.